Today I wanted to talk a bit about my science, about how we use genomics to inform bat conservation under climate change. But I wanted also to talk a bit about my experience, my personal experience as a queer person and a parent in science and academia. So the first bit of the talk will be the easy bit, talking about my science, and then let's see how the next bit will go. All right, so we're facing a biodiversity crisis, and really that's all linked to our impacts on the environment. And this biodiversity crisis is manifested in a form of mass extinction of species, but more so of populations. So if we look at the Living Planet Index, it's estimated that wildlife populations have decreased by an average 73% since 1970s. Now this is a massive decline that actually happened in my lifetime. But what we don't really know is what is the real magnitude of these declines. So what was the historical population sizes like, pre-industrial periods? But we also don't know that much about what are the consequences of these declines. So what's causing these declines? Well, we are causing them in our impact on the environment. So through aspects like fossil fuel emissions that's powering our computers that we're using and data storage, but also our travel around the world and everything we do pretty much. We're changing the climate, and this is resulting in increasing temperatures across the globe, changes in patterns of rainfall. And all this is creating conditions where are simply unsuitable for many organisms and forcing many species to shift their range and try to move and find more suitable areas. But climate change is not something that's happening on its own. We're also causing mass changes to habitats through habitat loss, habitat degradation, and that's all related to the land use changes that we're making, the levels of pollutions that we're causing in the seas, in the air, in the land. And what that means is that many organisms simply are not really able to easily shift their range and reach suitable areas. But it's also resulting in mass decline of species and populations because they don't have suitable habitats around them. Other impacts that we have on the environment is through overexploitation of natural resources. So putting this all together, it's quite a lot of pressures that are put there on wildlife. And how wildlife can respond to these different pressures really depends on the rate and magnitude of the changes that we are causing. But it also depends on intrinsic factors related to the individual organisms themselves. So this is related to their adaptive capacity. So can they adapt to these changes? Can they tolerate these changes? But also can they move away and reach other areas? So these are big questions that we're trying to understand. And what I'm going to try and show you in this talk is how we can use some genomic approaches to help us understand some of these issues. Okay, so when I talk about genomic approaches, what I'm talking about is trying to better understand the genetic makeup of organisms. So that code that's inside us, the genetic code that codes to what we are. So in combination with the environment that we grew up in, this genetic code will determine what we do, how we look, how we behave. And in fact, it determines a lot about how we respond to the environment as well. And through looking at our genetic code itself, we can understand a lot more about perhaps how organisms may be sensitive to future changes, but also it can give us a glimpse of how they may have responded to past changes. So what can genomic data give us? It can give us information about the conservation status of populations. It can tell us something about how the landscape is affecting connectivity between populations. So when we talk... It's all right, no worries. When we talk about connectivity here, what we're talking about is gene flow, so individuals moving from one place to another, mating with other individuals, and therefore whatever their genetic makeup is moving from one place to another. It can also tell us something about the long-term viability of populations, so can they actually survive in the long term through adaptation? And one of my big interests is actually looking at demographic history, so how the populations have changed over time. And this can help us understand the baseline historic population sizes. So what did these populations look like before we started impacting the environment in the way that we do? And the reason why genomic data can tell us this is because the genomic makeup of each one of us contains information of historic or ancestral genomes and the evolutionary forces that shape them. So through looking at individuals that live today, we can know a lot about what happened in the past as well. Okay, so in my group, the Global Change Genetics group, we use integrated approaches. So what we do, we combine genomic data with a lot of ecological research and different modeling approaches to try and understand how these global environmental changes are affecting wildlife. Now, a lot of our work focuses on bats, and this is because bats are amazing. They're really diverse in a lot of different ways. So there's over 1,400 species, so the second biggest group of mammals. They're also really diverse, as you can see, in their form, in their function. In their distribution, they're found pretty much everywhere around the world, obviously higher diversity around the tropics, but they are experiencing loads of different environmental conditions. They're very important for us as well, so they provide important ecosystem services in the form of pollination, seed dispersal, reforestation, and control of insect pest populations. But beyond that, beyond all the important stuff that we are actually benefiting from the presence of bats around us, they're also hosts of many zoonotic diseases. And for example, SARS-CoV pandemic has been linked to bats as well. So it's quite important for us to understand how these global environmental changes are affecting the movement of bats and potentially the pathogens that they have. Globally, bats are under threat, so over 25% of bats classified by the IUCN Red List as under threat, with over 50% of bat species having either declining or unknown population trends. So even the more common bat species have had quite severe population declines. So in terms of impacts of climate change, well, bats are thought to be quite sensitive to climate change, and the reason for that is because they have these very large membranous wings, and that means that they lose a lot of water through evaporation. So if bats are found in warmer and drier conditions, like in desert environments, they tend to have physiological adaptations that reduce the amount of water loss from their skin. And what that means is as bats are found in or experiencing warmer and drier climates under climate change, they will require some physiological adaptations to be able to survive. And indeed, if we look at what's been happening so far, we've seen mass mortality events of bats in Australia and in Southeast Asia in response to heat waves. Around the Mediterranean, we're seeing many events of bats suffering from heat stress. But even here in Britain, which is definitely never too warm for me, but there has been heat wave events where many young bat pups have been taken into care because their mums have left the roost because it was simply too hot for them to actually be able to stay there. So this is something that's having quite major impacts on bats populations. Okay, so how do we do it? How do we study the genetics of bats? So we always start in the field. And any excuse for me to travel and do field work, but we're going to catch bats across different places where they're found. It can be in forests, it can be in caves, it can be in buildings. We put out nets, so we put out different traps, we get the bats. Once we got the bats, we take little wing biopsy samples, so that tiny little circle over there, and we store it with some buffer. And this is what we're going to use in the lab to extract DNA, so their genetic code and genetic material from these bats. And then we use different sequencing technology to be able to, in a way, take this information into something that we can read and analyze. Okay, so I'm going to start by talking about how we can use these kind of genomic approaches to identify population declines. And I mentioned how we've seen global declines of wildlife populations around the world. I mentioned within the context of bats as well, how many of them have had mass population, or their populations are thought to be declining or unknown status. So anecdotal evidence, and when I say anecdotal, I mean anecdotal, suggests that bat populations in Britain have declined dramatically in the past few hundred years. Now this is in response to habitat loss and persecution. However, this is just anecdotal evidence. In most cases, we don't actually have any empirical evidence to support these statements of bat declines. And this is quite a big thing, because if we try to advocate for bat conservation, when we're trying to say, look, our impact on the environment has really affected bat populations, that's why we need to conserve them, we need to be able to show that it really had an impact. And one of the reasons why we don't have this evidence is because monitoring efforts, or standardized monitoring efforts of bats, only started around 30 years ago. So what we're really seeing is stuff that only happened since the 1990s. And if you looked at that graph of global wildlife population declines, you can see that there's been mass declines happening before that. And this is something that's been a big issue for the Bat Conservation Trust, so the main conservation organisation advocating for bat conservation in Britain or in the UK. So they contacted me to try and develop together approaches where we can better understand what historic bat populations used to look like and have this kind of evidence that they can show that bat populations have declined, including populations of more common bat species. But to make our lives easier, we decided to start working first on a rarer bat species, so the western barbastelle bat, Barbastella barbastellus. So this bat is a species of European conservation priority. It's closely associated with old-growth forest habitats. So this is a kind of habitat type that's really been declining across Europe. It's thought that their population is declining. However, we don't actually know anything about the population sizes. And the reason for that is because these bats roost in trees, so it's quite difficult to monitor them. So they tend to move around from one tree to another quite often, so it's quite challenging to know what's really happening there. So what we did, we generated genomic data sets, and our interest was to, first of all, identify whether there have been any population declines, try and put a time of when these happened, and also to try and identify what are the drivers of the decline of the species, if there are indeed declining. Okay, so first we started by looking at different landscape drivers of genetic composition. So this is a genetic makeup of our different colonies that we sampled. And what we found is that levels of genetic diversity increases with a percent of broadleaf forest cover around the colonies. What that means is that areas that have a lot more forest around it, the colony has much higher genetic diversity. And that's probably linked to the fact that it can support higher number of individuals, these areas. Okay, so then we tried to look at how the landscape is affecting connectivity between our populations. And when we talk here about connectivity, we talk about genetic connectivity. So that's the movement of individuals from one place to another and the mixing of the genotypes. And what we found is that genetic connectivity between our different colonies is impeded by artificial lights at night. So if you have a lot of artificial light at night, these bats simply are not likely to move through these areas. But it's facilitated through woodlands, but also riverine habitats. So we know these bats tend to commute from one place to another along rivers and use a lot the riparian vegetation around rivers for foraging. So putting this together, we can see that habitat loss, and particular loss of forest habitat, has resulted likely in a reduction in genetic diversity and connectivity in these bats. Okay, so next, how do we look at evidence of historic population declines? So as I mentioned, the genome of an individual contains information on ancestral genomes. So what we do, we look at the genetic makeup of current populations, and we try and compare different scenarios of what may happen in the future. And we use an approximate Bayesian computation modeling approach to compare these different scenarios and see which one of them best fits what we currently know about the genetic composition of our bats. So we compare a scenario of there's been no change. So over time, over the past few thousand years, there's been no change in population size. And then we compare it to different scenarios of either population increasing or decreasing, and different populations experiencing different things over time. So the best scenario, the best supported scenario we got in this case was one of our population declines by 99% in both the southern and northern British populations. This is really major population declines that we're talking about here. And this declines was estimated to have happened, or at least started, around 300 to 500 years ago. So when I first got that data, I was like, that's a bit old. I thought it's going to be in the last 100 years, I don't know, industrialization. But then we looked at it in a bit more detail, and that period is the 15th to late 17th century. And this is known in European history as the Age of Discovery. So this is where you had the first colonial expeditions going around the world, colonizing the world. So to be able to support these colonial expeditions, they needed very extensive shipbuildings. To build these ships, what they needed is very large quantities of these outside mature oak trees. And indeed, if you look at historical records from these periods, you have quite a lot of records talking about this loss of woodlands and loss of forests, and especially loss of these larger trees from the landscape. Now, these are exactly the same trees that are used by the barbastelle bats for roosting. So what we can see here is perhaps a situation where the decline of these bats may be linked to the loss of their maternity colonies in these trees that were felled down to support shipbuilding for colonialism. So as I say, colonialism, also bad for bats. There you go. Okay, so we published a paper on it, great, about 10 scientists read it, very important. But then, is it really going to affect the conservation of the species? So what we did, because I work closely with conservation organisation, we worked also with Natural England. And we used this information to help define what Natural England calls the favourable conservation status of the species. What this means is where do we want to reach our conservation efforts? So what does a thriving population look like? And given that we have this information of what past thriving populations look like, it helped us really be able to set targets for conservation. So where do we want to go to in the future, and what is the current status? So also all the information about the impacts of land use change helped us also develop conservation measures as part of this as well. So it's just to show how, it's great to do science, but it's really important to also try and apply it and really change things, and especially if you're interested in conservation. Okay, so I'm going to move now to talk about how we can apply genomic approaches to assess vulnerability to climate change. And this is different framework and approaches that we've been developing over the years. Now, if you think about the impacts of climate change on biodiversity and trying to address it in a way, we need to first know which species are actually going to be sensitive. But once we know which species are sensitive or vulnerable to future climate change, another thing that we need to know in terms of conserving them is to know which populations do we need to focus on. So where should we focus our conservation efforts? And then what conservation efforts should we actually do to try and help the species? So this is the kind of questions we try to answer. So the first question is which populations are actually under threat from climate change? So how do we know it? First thing we look at is exposure. So will they be affected by future changes? Next we look at sensitivity. So can they cope with the changes that are happening? And finally, movement, or can they actually move away or get away from the changes? I'm going to show you some examples of how we use these kind of approaches on three bat species. The first is the grey long-eared bat, Plecotus austriacus, very close to my heart. I did my PhD on this species. It's found across southern Europe. You could see, in Devon, definitely, but just in the southwest of England. So this is really the northern edge of its distribution. And it's one of the rarest mammals in the UK. Next case study that I will put into there as well is this two cryptic Naterere myotis bat species. So these are two species that look morphologically identical, but genetically they're different. Myotis escalari is found around Spain and Portugal, and Myotis crypticus is found around the northern parts of Spain, southern France, and Italy. Okay, so let's start with exposure. How do we study how species are going to be affected by climate change? So this is commonly done using what we call species distribution models or climate niche envelope models. How it works is we have information of where the species is found, so location records. We combine it with information about the environmental conditions where the species is found, different statistical and dynamic modeling approaches, and then we project where suitable conditions are found across a wider area. Now we can also do it by using climate change scenarios and project where suitable conditions will be found under future climatic conditions. So this kind of information we use extensively to project extinction risk to species under climate change, coming up with very large proportions of species that are likely to be affected. Okay, so looking now at the grey long-eared bat, here we see the condition under present conditions. So this is a range of suitable conditions for the species. Orangey, yellowish, greenish areas are suitable, bluish areas are usually tend to be less suitable for them. You can see under present condition, this is quite a southern European species, and only part of Britain is suitable for them. Okay, so then we projected to the future to what climate change projections are to the end of the century under a high emission scenario. What you can see here is sort of like a northwestern shift in suitable conditions. Suddenly the whole of Britain and Ireland is highly suitable for this bat. But if we look at the areas where this bat is very common, so especially particularly around Spain and Portugal, many areas are becoming climatically unsuitable. And we can use this kind of information to identify these populations that will be found under unsuitable conditions in the future. So they're likely to be high exposure to future changes. Okay, so next we're going to look at sensitivity. So can they cope? Now there's different ways how to look at sensitivity. You can look from a physiological perspective and whether they can tolerate these higher temperatures. In our case, we look more from a genetic perspective. So have they got adaptations that means that they can survive under warmer and drier conditions? So we do it through looking at the current adaptations to the local conditions they experience. And what we're looking at is two environmental variables that are very important for the survival and reproductive success of bats and will also change under climate change. So it's summer temperatures and summer rainfall. So we use here genotype environmental association analysis. So what we relate is the genetic makeup of each individual bat to the climatic conditions that are found across their range. Using that, we can identify areas of the genome that are associated with particular conditions. So for example, with hot and dry conditions. And then we divide our individuals based on their genomic makeup into those that are in red associated with more hot or dry conditions, those in blue that are associated with cold and wet conditions, and those sort of intermediate individuals in the middle. And then we can plot this on the map and we can look at our different populations. So we have here these populations that are found in hot and dry conditions, and we have over there those that are associated with more cold and wet conditions. So if we think about sensitivity to climate change, which ones are likely to be more sensitive? Those that are here in blue and grey are likely to be a lot more sensitive, less likely to cope with future changes. Okay, so finally, next we're going to look at movement. So can they actually get away? Now, the ability of organisms to move across the landscape is really likely to be impeded by landscape barriers to movement. And these can be natural barriers like a river, a mountain range, or an island surrounded by sea. And very terrestrial in my perspective. So I suppose from somebody else's perspective, it will be the land that's a barrier. But it can also be anthropogenic barriers. So it can be urbanization, it can be deforestation, anything like that. So what we do here, we look at their genetic makeup of our individuals, but what we're interested in particular is differentiation between different populations. So if the populations have similar genetic makeup, that means that a lot of individuals are moving between them. If they're very different, that means that we don't have that much movement of individuals between them. And then we relate it to different landscape variables that are likely to affect the movement. So these can be things like land cover, habitat suitability, can be temperatures, altitude, forest cover, big thing with bats, or light pollution, for example. Okay, and once we find what really affects the movement, say in the case of the grey long-eared bat, for example, this was habitat suitability. What we can do, we can plot the potential movement density given the impact of the landscape on genetic connectivity between our populations. So yellowish green is high movement potential, blue is low. And what we can see is quite good movement potential here across the range and between the different populations. But then we can also project it to the future and the future conditions. And then what we suddenly see is some of our populations are becoming isolated under future conditions, and there actually is less or more isolation happening between what's happening in Spain and Portugal and Britain. So these kind of populations that become isolated under future conditions are less likely to be able to shift their range and move to suitable areas. Okay, we can also use this kind of approach to try and ask a question of how can populations survive where they are. So if you think about bats, they're very long-lived, slow reproducing, so there's less chance that suddenly a mutation will happen that means that they can survive under future conditions. What's more likely is if we currently have a population that's maladapted to future changes, so they're adapted to cold and dry conditions, and we know the area will become warmer. Sorry, cold and wet condition, we know the area will become warmer and drier. How they may be able to survive is if you have individuals from a population that is more adapted to warmer conditions moving up there. And as they move, they bring in their genetic makeup, they interbreed with one each other, and hey-ho, there'll be some individuals in that population that will be able to survive. But this all depends is whether the landscape allows them to actually move around and connect between populations. So we do the same kind of analysis, but here just focusing on these two Myotis cryptic species. In their case, they're closely associated with forests, so forest is the main variable that affects their movement across the landscape. And if we look here at these movement density maps, we can see that we have some areas where there's high movement potential between our adapted to hot and dry conditions populations in white to those that are adapted to cold and wet conditions in grey. But we do have some populations there more at the north that are not likely to receive this kind of genetic variation that are associated with how conditions are going to change under climate change. So landscape connectivity is something quite important that may affect the survival of individuals as well as their ability to move from one place to another. So the next step is to put this all together. So we have information exposure, we have information sensitivity on their movement potential, and then we want to put it all together into population vulnerability index that can tell us about how each population is vulnerable and likely to be affected by future changes. So I was working with Chris Barrett at ITIV in Germany, and we were developing together an informatics toolbox to put this all together and make it a lot easier for people to do it in a more automated way. So let's have a look at how it works. So here you have from low vulnerability on the left to high vulnerability on the right. So these low vulnerability populations will be ones that are found in areas where less exposure to future changes. So conditions will stay relatively the same. In terms of sensitivity, they're already adapted to warmer and drier conditions, so likely to be able to cope. And in terms of movement, can easily move around from one place to another. So these will be our low vulnerability one. Then at the other end, we have the high vulnerability ones. So they're found in areas where conditions are going to change very dramatically. They're likely to be sensitive because they don't have adaptations associated with warm and dry conditions. And they perhaps cannot move very far because the landscape is going to change and make it more challenging for them to move. And in the middle, we have a situation where the climate is changing, but our populations may be either adapted or they can move to different areas. Okay, so that's the research side of things. What about the applied conservation side of things? So we can use this kind of information also to inform conservation. And conservation management can move from least intensive to more intensive intervention. So these low vulnerability populations, because the area is still going to be suitable, they're adapted to future changes as well. What we want to do is make sure that these populations are as large as possible. So we want to improve the habitat around them and make sure they are protected and can be a source that can go to other places and help other populations survive. If you think about the really high vulnerability, quite challenging. Perhaps the really realistic solution is to physically take them from one place and relocate them to another, which is too costly if you really think about the extent of climate change, the amount of species that will need intervention. So a lot of the time where we want to aim our conservation management is more at the mid-vulnerability populations, where if we increase landscape connectivity, we can enable them to move to other areas, but also to receive gene flow or individuals that are coming with genetic makeup that will enable them to survive. Okay, so some take-home messages to put it a bit together. Hope I showed you that we really need this kind of integrated approach to tackle these kind of biodiversity and climate change crisis. In the end of the day, it's a very interdisciplinary kind of area, and to better understand how things are happening, we do need to gain as much information as we can from as many sources as we can. A lot of my work has shown that we are already seeing severe population declines in bats, and it's linked to the loss of, in the case of these species at least, loss of mature native forests. And this means that when we think about conserving them, we have to take into account how much they've actually declined over time as well. I also showed here how we can use the genomic approach to improve the climate change vulnerability assessment and better address our conservation efforts. But all in all, message that I always want to say is if we want our research to really make a difference, we have to work with conservation practitioners, we have to work with policymakers, but not just at the end. So we finish the research and they're like, yeah, hello, listen to me, I've done something really important. But it's something that we must do really from the beginning and make sure that they're involved in co-developing our the project, co-designing them, and that the projects that we do really address the conservation need. Okay, so just to mention many people, and even more than that, who contributed to a lot of this work. So this is a science talk bit. I thought I'll spend like 5 minutes or so just saying a bit more personal stuff, which is obviously a lot more challenging because talking about science is easy. Talking about myself feels very self-indulgent. But I thought I'd say a bit about my experience perhaps as a queer person in academia and as a parent in academia. So I'll first perhaps start that talking about myself as a parent in academia seems a bit weird to me because I've only become a parent at an, older age, I suppose. I never saw myself as a motherly type or as a parent, and it's only when I reached age 39 and I thought, it's now or never, I sort of realized that I'll regret more not having a child than having a child, so I decided to have one. So it's not necessarily the type of role that I thought I'll see myself as. Yeah, so that's just a clause about being a parent in academia and me talking about, which feels a bit weird. But I suppose all of it comes to the fact that I never really conformed to gender norms. And ever since I was quite young, I never saw myself as really belonging to necessarily to the gender I was born in. I'm a woman. And I'm very happy with being a woman in the sense of I have no problem with my body. I have no problem saying that I'm a woman, and I love having periods because it's part of the cycle of nature. However, I never really identified necessarily as a woman. And it's been all right for me all my life. So I had a lot of support from my parents and all my friends and everyone around me who never really treated me in any way but who I am. So although my parents did try to send me to ballet lessons when I was younger and make me wear dresses, they pretty soon realized that that's perhaps not who I am. But they always really encouraged me and never put any barriers in my way. So when I was 5, they sent me to chemistry club, after school club, which seems a bit weird, but I loved it, and ever since then I loved chemistry. But they also always encouraged me as doing science, doing math. I have 2 younger brothers, and I was never treated any different from them in a sense that I was always treated as being the older one. I'm the stronger one, I'm the more adventurous one. And I know this doesn't sound amazing. However, over the years, talking to a lot of other women in academia, I realized that that's not the experience that everyone has had. And a lot of women have been brought up in a way where they were taught that they perhaps are not quite as good in the science side of things, and perhaps that's not really where they should see themselves. And even if they weren't taught that directly, they were made to feel like that is where they are. So I think I was very lucky to have parents that did not put this burden on me and didn't gender stereotype me. I was also very lucky to, throughout my academic journey, to have a supervisor that really supported me and really promoted me. And after that, being in research fellowships, I was always very promoted through what I did, and I was never actually treated any differently or been put or boxed in any place or stereotyped or put into any gender norms. And when I realized that that's not the experience that everyone has is when I was part of mentoring circles for early career researchers when I was doing a fellowship in Bristol. And a lot of the women that were there actually had a lot of baggage that they brought in with them, and that resulted in a very strong imposter syndrome as a result of how they were gender stereotyped throughout their lives, which I thought I was very lucky not to have gone through that. And when I was pregnant, that's the first time that I actually realized what it is like. So all my life I was never necessarily treated as a woman, I was always treated as a gender neutral person. But when you're pregnant, it's quite different. You're very obviously a woman, and everyone, how they treat you, how they talk to you, what they talk to you about is very different. And it's something that I found very challenging, but it opened my eyes to the fact that perhaps other people have had different experiences from me. But having kids is something that also opened my eyes to the importance of what we want to pass on to the future generations and the type of core values that we have. And I always brought up my kid in a very gender neutral way without making them feel like they have to fit into any gender roles, and making them aware of alternative ways of life, making them aware of different forms of families, different gender identities, and how it's important to accept everything. And through the eye of my child, I can also see how I perhaps was when I grew up, and being less aware of these issues, given that I grew up in a time where it wasn't even talked about, these issues. And I can see with my child, who is born as a girl, but doesn't necessarily identify as a girl, always used to, when much younger, used to say, I'm a boy. Nowadays, a bit more, just happy not to mention gender or be gender neutral. But it really opened my eyes to the fact that perhaps I was the same, but back then I didn't have the language to even say these things. And it's really great to see how other kids in the school accept my child as they are and don't necessarily try to stereotype them. So finally, in terms of being a parent in academia, it's a challenging thing. And being a scientist is something that takes all your time. It takes all your thoughts. It's something that you're very passionate about. So you put a lot of effort towards it. And it's not just the science that really takes a lot of your time. It's also the education aspect of it, because you want to pass on knowledge. You want to mentor younger people. You want to really make change. In my case also I'm involved being the Director of Equality, Diversity and Inclusion in my department, because this is something that I truly believe in. So all these things have a lot of pressures on your time, and you've got to always remember that that is really important, but it's only a job. And you've got to remember you've got to put your family, you've got to put your life, your personal life and your mental health and yourself first. So that's something that I've had to learn over the years and make sure that I don't leave my child as a second option. So when it's my time with my child, it's my time with my child, and I put my work away. Because otherwise they're never gonna, they're never gonna have any confidence in themselves, doesn't matter what you say to them, if you don't treat them as if they're the most important person for you. So I think that's it. That's all I've had to say. Thank you very much.